we are going to be having our first panel of the day. And why don't I just welcome on the moderator here, Jason Kolbert from Dawson. James, is it Kolbert? Is it Kolbert?
Kolbert, you got it.
Kolbert, awesome. I will let you introduce the guest, if that's okay, Jason.
Sure, my pleasure. First of all, Mark, great to see you. Great presentation, as always, incredibly thorough. What I hope is that you won't mind repeating some of the things you just said. I've asked all of the CEOs to talk a little bit about their company and introduce themselves, the company, the ticker, the market cap, cash on the balance sheet, and when they think the next catalyst is. And then we'll get into some Q&A. As an introduction to everybody, I got to tell you, it's an interesting time, right? COVID is a black swan. It's not something that anybody specifically predicted, although many people believe that we were susceptible to a either natural or terrorist-like event for a pandemic, but now it's actually happened. And there are some good news about COVID is that this is a good dry run because it may happen again. What's interesting is the resources that are available in the world of biotechnology to treat it. And I think the first step in COVID was to be able to diagnose it. The next step was to see, can you treat it? How do you prevent people from cascading? And of course, the ultimate goal is how do you arrest the spread? And I think arresting the spread occurs with vaccination. The problem historically has been that vaccinating against a coronavirus is very difficult. We don't have vaccines against the common cold because they change a lot. But because of the uniqueness of this specific virus, it seems that there were conserved regions in the way the virus is built and that vaccines could be created. And the market moved very quickly with a new type of gene-based vaccine technology, identifying a specific region of the vaccine that doesn't change. This is a little bit like identifying a person by their nose. Not by their ears, not by their hair, not by their eyes. But when you get up close, the nose can be used to identify. It's a different type of vaccine than what we've seen in the past. It's very clear to me as an analyst that we will see second, third, and fourth generation vaccines. And I think that's very relevant to this panel. It's also extremely relevant when we talk about, do you need a booster for COVID? By the way, I'm pro-vaccine. I've had both of my Pfizer shots. The real question is, at what point would I need a booster? Do I need a booster? My B cells, my immune system should remember the virus. They should be able to activate my T cells to kill the virus. But what is the level of my circulating monoclonal antibodies to prevent an infection from taking hold? How do you know that? Because a binary test, whether I have or don't have COVID, doesn't provide a lot of information for us. So I think that's a good place for us to start. When we move beyond traditional binary diagnostics and we think a little bit like a treating clinician, what kind of information does a clinician want to hear, want to know? So let me introduce one of my favorite companies, Aditxt. Amro Albanna is the CEO. He's the founder of the company. And I think COVID is a little bit of a test case for this company because I think success in this company has much broader implications, not just to COVID, but what additional immunological information would you want to have as a treating clinician in order to treat your patient, let's say if they had cancer, or let's say if they had Crohn's disease or another immunological related disorder. I'd want to know exactly what is the status of their immune system. So with that as a lead-in, let's talk first a little bit about Aditxt. We're going to keep our initial comments for each company to 5 minutes, and then we're going to go to a moderated Q&A format. Amro, take it away.
Jason, thank you. Good to see you again. So Aditxt, we are a biotech company. We're focused on commercializing new innovations that take fundamentally different approach to treating and understanding the immune system. And Jason, you summarized it very well in your intro.
Amro,
but before
don't forget. Name,
yeah, I was
ticker.
going to say. So again, my name is Amro Albanna. Sorry, My name is Amro Albanna. I am the co-founder and CEO of Aditxt Inc. Our symbol is ADTX. As of our last filing, second quarter of this year, we had $8.6 million on our balance sheet. And we did another round of financing, $11 million that we announced in Q3, primarily for the purpose of a potential acquisition that we're looking to do in the antiviral space. So the DNA of our company, no pun intended, we are a biotech innovation company. Our mission really is to innovate and commercialize various biotechnologies that fundamentally or take fundamentally different approach to precision medicine when it comes to the immune system. So what does that mean? In one area, we are developing new therapeutics, new class of therapeutics, where we could potentially retrain the immune system to address organ rejection, autoimmune diseases, as well as allergies. And that's really how the company started, Jason, which really led us to the second platform that we have been working on, which is Aditxt, a way to understand the immune system. Because here is our belief. We really cannot begin to speak about precision medicine and precision type approach to our health unless we understand someone's immune profile. We certainly were not planning on COVID. We did not anticipate COVID, just like everybody else. But when COVID hit, we recognized that we could use Aditxt, our platform for profiling an immune system, to begin to understand how we each react differently to COVID and what type of immune response. And maybe I'll just kind of end it with this. Early 2020, when COVID started, the world was focused on controlling infection. Everybody was focused on protecting society from each individual. So we had to wear masks, we had to do social distancing, we worked remotely, we stopped flying, so on and so forth. But during that time in 2020, we actually went public and we said, while everybody was saying when vaccines become available, things are going to go back to normal, we predicted that when vaccines become available, the focus is going to shift from infection status to protection status, because ultimately the way forward, we each have to understand our personal immune profile to make decisions in terms of treatment and how we move forward on, including booster shots, which is obviously becoming more and more of a discussion and a debate as we move forward. So that's Aditxt. We're applying it to COVID-19 and many other applications in potentially oncology, autoimmunity, CNS, and other areas.
Amro, when we talk about booster shots, what is the immunological profile of somebody that would clearly need a booster shot?
So, Jason, this is going to be a major question, and our goal here as Aditxt is to really to get people, and especially clinicians that are practicing out there, to give them objective information to make that decision for their patient or for each individual. Because you've seen the debate, you've heard the debate in the last 60 days as far as booster shot, whether or not someone should take it. It is what I will say first. I certainly hope, and we certainly believe that booster shots should not be provided based on a population type approach. Everybody gets it, or everybody doesn't get it, or everybody gets it at a certain time. Look at immunocompromised. Some of the immunocompromised patients, as we're seeing in our data, even with a booster shot, they're not developing immune response. So that may not be the best approach. I'll use myself as an example. I got COVID early 2020. I didn't realize it until I got Aditxt. I received 2 Moderna shots, so my immune response is off the chart. Do you really want to give someone a booster shot, someone like myself, a booster shot? So the approach really moving forward must be based on individual immune response as we're making decisions about booster shots or even course of treatment.
Or if I can just add on one part, if you've already had COVID, do you really need to have the vaccine? What would the Aditxt score tell you? And so is natural immunity enough, or should you get the vaccine?
So this is obviously an area of major interest, and you've seen some of the studies. In fact, you've seen the study that just came out of Israel when it comes to breakthrough infections for those that are naturally natural induced immunity versus vaccine induced immunity. And Jason, the stance we're taking with Aditxt is we're not going to practice medicine. We're not going to tell someone whether or not to take vaccine or whether or not they have this or that. What we're actually saying is this: whether you had COVID, whether you were vaccinated, or even if you had COVID and vaccination, it is best to understand your immune profile so you and your physician can make that decision. And frankly, all of us are continuing to learn more and more on how our own immune system is responding differently to COVID, and of course, other future viruses. As you mentioned in your intro, this is certainly not the first, and it won't be the last.
So I want to push this just a little bit more, which is, once you've had COVID, if you have an Aditxt score, the Aditxt score may show that you have immunity, but that if you get a COVID vaccine, you may actually get an even enhanced level of immunity. And that was your experience.
That's my personal experience, correct? Because you can look at it as, to put it simply, of course, when you get the virus naturally, we're not sure exactly how we can count the number of exposures. But look, if in my personal case, I received, let's just say, 3 exposures, one time with COVID and twice Moderna. So that increased. I initially, before I got the vaccination, I was already showing immune response. Robust immune response. And then I took Moderna because I had to travel internationally. And that, like I mentioned, got my immune response off the chart. So pretty high immune response in my scenario.
Okay, so one last quick question for you, Amro, which is, it's a small company, sounds like a really good technology. I haven't heard of a lot of other companies that are able to immunologically profile very specific markers to determine not just whether you're immune or not, but where your immunity is, what the markers are. And by the way, I've had the Aditxt score. Thank you very much. And it was fascinating to look at all of those different markers. So my question for you is, what's it going to take now to commercialize on the COVID side? What does the revenue ramp look like? And I'm not asking you to project future earnings, but just in broad terms, what are you doing in order to get this technology commercialized, which will ultimately be a driver for the stock?
Yeah, no, great question, Jason. Look, we've been public for a little bit more than 12 months, about a year. And you probably heard me say this before. We are a great story. We have great innovations, but I'm pushing the company to get into growth story, which is commercialization. And what we've been doing in the last 12 months is we've built the infrastructure required to actually commercialize Aditxt score. So that included opening a large-scale immune monitoring center in Richmond, Virginia. That was early July of this year. That included, of course, complying with regulatory requirements. We are a lab-developed test. That's a key word for us. That's a key word for us because we fall under CLIA and CAP regulatory bodies. We had to understand the private and public insurance. We identified at least some of the CPT codes that we can at least share with potential partners. They obviously have to make their own decisions on that. We applied for Medicare. So we've done all that to provide a commercialization infrastructure to deploy Aditxt score. So from here, our strategy to go to market is to partner with channel partners. That's what we call hospitals, independent physician offices, even other CLIA labs, where they send us the samples, we process the samples, and we send them back Aditxt score for their own clients. That's our commercialization plan. We've been successful in doing that. As we go around the country and we share Aditxt score with various chief medical officers and other physicians, they're really positively surprised that there's such a tool that exists, and that's our plan. Finally, on your last question, yes, this is certainly not a forecast, but to give you an idea, right now we are Generally speaking, each Aditxt score is anywhere between $80 to $90. That's our revenue for each Aditxt score. So if we end up doing $1 million, that becomes $80 to $100 million in revenues, which comes very significant for a young company like us. If we move up to 10 million, which is, that's the scale that we've built in Virginia, in Richmond, Virginia, well, that would mean $800 to $1 billion in revenues. Certainly, those are just the general numbers, and that's not an indication of a sales pipeline.
Understood, but you have capacity to go up to 10 million. At 1 million, the company would be wildly successful, by the way, if you pulled in those kinds of revenues. Really interesting story. Obviously, I'm an analyst. I'm covering this company. I'm covering Dyadic. I'm covering Sorrento. Anybody who'd like to see the research that I'm writing, go to the Dawson James website. By the way, these CEOs can tell you, I won't hesitate to challenge them if something they're saying doesn't make sense. I'm a very tough analyst. I'm not just rating stocks to do business with them. I'm putting it where I really mean it. And they're all buy rated. I all think they're really great names. Thank you so much, Amro.
Thank you, Jason.
So I have a challenge mentally. Do I transition to Dyadic and talk a little bit about kind of their vaccine platform? But I think it'd be more interesting to go to Sorrento. And then as we talk a little bit towards the end in Sorrento about why Sorrento is working with Dyadic, I think Mark will set you up to kind of close the panel, okay?
Great.
Okay, so Henry, one of the things that Amro mentioned and that I kind of mentioned is that this is a new vaccine technology, mRNA, and my understanding is that it's essentially going after one conserved region. I see a lot of flaws in this. In other words, I see it as a first generation technology. Scientifically, I know that you could talk about anything, and we'll get into the diagnostics, we'll get into the therapeutics, and we'll get into the vaccines. But I'd first like you as a scientist to opine about what you see in terms of the vaccines after you finish your introduction, name, ticker, market cap, balance sheet, and talk with me a little bit about, is this a first generation vaccine technology? Will we see second and third generation?
Thank you, Jason, very much for inviting to the panel. So, as you know, Sorrento is listed on the NASDAQ. The ticker symbol is SRNE, and we have a market cap of about 2.2 billion, and we have cash and the investment in public and private entity all coming together in the few $100 million range. And as answer your question about the messenger RNA, this is the first generation right now. As you know, the mRNA technology is evolving very fast pace. As you know, you can tell this is a first generation. Reason is Sanofi just bought a company, Translate, and that company they bought about 1 month ago. They invested firstly years ago about the 450 million. Then last month they bought the company for 2.3 billion, and I believe it's announced yesterday or the day before they scrapped the whole things because it's a mRNA, it's unmodified. So that tells you this technology evolving so fast that even the big players over there right now is reactive, and I believe that the one that unmodified mRNA could be difficult. That's where the Sanofi scrapped the whole few $1 billion acquisition and stopped the program in the unmodified mRNA vaccine. And you need to keep up with the advance of the technology. The first generation mRNA using Washington strand or the Wuhan strand, and that's the spike protein. Now, the issue is the spike protein. You have some cleavage site in the middle that potentially cut the spike protein into S1, S2. With the S1 leaving the memory and floating in the bloodstream, that may cause some side effect. So you want to change that. So the first generation, none of them changed the mRNA structure. That's problematic potentially because that's where potentially a side effect kicks in. And secondly, the mRNA, you have a formulation. The formulation right now is using lipid nanoparticle. The lipid nanoparticle, firstly, the lipid they're using, they may have some IP issues, right? And then lipid nanoparticle itself, a first generation with some pegylation in it. And, you know, there is a lot of drugs using pegylation. You generate an anti-idiotypic, or anti-drug antibody, ADA, we call that, potentially against the pegylation. So you got to have a problems, because then you got to have a systematic, side effect or certain specific organ side effect. Now, this is improvement.
I want to interrupt you and kind of translate what you just said, which is the current mRNA vaccines, the way they work, and by focusing on the spike protein and where they kind of create that spike protein, that there may be elements of that have some toxicity associated with it.
That's a potential concern. That's a potential concern. Yeah,
and,
you need to perfect it.
And the formulation, in order to get this gene therapy right into the target cells, that formulation itself could be immunogenic and any problem, particularly when we talk about second or doses or repeat doses, right? Exactly.
That you create the antibody against the drug antibody, or antibody against drug, And that's very dangerous,
Henry. So what is the solution to that? I mean, as a scientist, and anybody who speaks with you very quickly understands that you're a scientist, right? You're almost you have multiple hats on. So just sitting back from a big picture point of view, what is this? Why can't we use a more traditional vaccine?
Yes, that you can, but the mRNA, what you needed to do is perfect it. So you perfect the mRNA sequence itself, which we did, and then you perfect the formulation. Then you're avoiding some of the formulation causing harm if you do repeated dosing, and which we did too. And then you have a formulation you can be stored in the room temperature. That's very critical. But then you can lyophilize, ship around the room temperature. That's going to be much more usable worldwide, especially for developing countries with no freezers, right? Of course, if you want the traditional antibody, which is a Dyadic, kicks in with the protein, which is RBD based, subunit RBD, which is right to the target, which is the one binding to the receptor for viral entry. And the protein is much smaller. It can be made with the C1 technology that Mark and the Dyadic developed it. It could be a tenfold of quantity than the traditional chill cell based antibody production type. So that's why we're very interested in working with Dyadic. The reason is that they give you firstly much smaller unit to work with, much higher production, much easier to transport, because that you can transport the RBD protein any type of form. You can be in a liquid form, refrigeration, or you can be dry form in the room temperature.
Is there a reason to believe that the RBD protein, which is not a genetic sequence, right? It's not the mRNA vaccine primes immune cells to kind of make spike protein, and then the immune cells recognize spike protein and they defend against it. The technology you're talking about, how does that work?
Yeah, that protein basically they are presenting to the antigen presenting cell, APC we call that, and they eat up the RBD, they show up and they present, and you got the immune cells, both the T cell and the B cells responding to it, and the hope is getting the memory long-term type of cells already created, but these generate very strong immune system because you can test it. You can test in animal very quickly, and you see the RBD that the Mark sent to us. You can generate very strong immune response. You can also testing whether these immune response against different variants. For example, if you use the RBD from Washington strain, are they against the Delta? Or what if the next one, are they against the South Africa? So that type of stuff, you can all test it before you get into human. And when you get into human, you can test it. Now, the beauty of this one right now, although in the North America, you have a large people quantity already been vaccinated. However, but when you do universal boosters, but you don't want to use the last year's flu vaccine as a universal booster for current year's flu strain. The same thing. Why are you using RBD or the strand mRNA from a Washington strain or the Wuhan strain, which is a lot
I could not
effective.
agree with you more. I've tried to follow the logic at the CDC, at the FDA, and even from the White House, and I'm just really struggling because for me personally, I'm trying to understand boosting people with the same exact sequence a third time to create circulating monoclonal antibodies. Doesn't make sense, particularly if you could do a variant or a multivariant booster, right? That makes sense to me.
That's the plan we have as a multivalent. So you can test the vaccine, you generate either mRNA or the RBD. You see whether they're against all of the other variants, because it can be tested very readily, very quick.
Henry,
we're talking about within a week we can test it.
Give me a timeline on the vaccine program on when we can expect some news.
So from the Dyadic RBD base, Mark already has the tox data, and we doing the IND package. So we're going to be able to, together with Mark and some of the other folks, we can submit to either the FDA or some other regulatory agency around the world. We already talked to German regulatory. They like the approach. And also you can do multivalent. That means you're putting multiple RBD into a same vaccine. As long as you have the tox data to back up, they don't need you to do another tox study. Okay, so that's beautiful. So mRNA, we're talking about, we're making the product multivalent. We're gonna go to Mexico and test it over there because the vaccine rate is very low. So you can do de novo. Meanwhile, you can actually, in the US, you do a universal booster. So you're using a multivalent, let's say you're using Delta vaccine or the South Africa vaccine, you're against the Delta, you're gonna have a superiority against the Washington strain, which is the Moderna, Pfizer, or J&J. We actually have a test, which is Amro said, we have a test to test whether you did the vaccine with the J&J. Do you have an antibody against it? I mean, if you have one shot J&J, in 2 months, you don't detect any antibody. And
when can we see this vaccine in a clinical trial?
My hope is both the RBD as well as the mRNA multivalent in the first half of next year in human. Hopefully, we can finish complete enrollment too.
Wow. Okay. And just, I don't want to, I'm almost out of time for you on Sorrento, but can you give me 1 or 2 minutes on the diagnostic side and on the therapeutic side? Just give me a mention on what you have and kind of what the events are we should focus on.
Yes. So the diagnostic side, we have a COVID stick. EU approved in Mexico. We're commercial launching the product already. You will see a big launch effort with multiple sports events going to be using a COVID stick. And we are talking about the double millions tests going to be marketed in Mexico and some other Latin American country. We are going to get
real-time point-of-care tests.
Yeah, it's a point-of-care tests. And we have real time, and we have Brazil going to be approval hopefully shortly, and Canada we're waiting for final words within a month, and the US we're very active with the FDA. Hopefully this time we're going to get the approval. Hopefully with the data we can get approval. With this, the demand is humongous. You have the last year $5 per tests, and the Abbott supply 150 million. This year $13 tests. Abbott only supplies 3.8 million. That not even last for a single day. So that's huge demand shortage right now. If you detect positive, we have a COVID drops. You do nose drops into your nose, preventing you getting into deep lung infection and get into hospitalization. That's the goal. We're working with multiple states right now. Want to do a few thousand patient different states so we can get the COVID drops in place too.
And COVID drops is a monoclonal antibody mixture that binds to free virus.
Yes, they binds, blocks the replication. This is a humongous demand right now, and there is no enough supply because the Regeneron requires 1.2 grand. It's not enough to make. Yes, it's not enough. And last quarter they did close to 3 billion. This quarter they just have an order from the government 2.9 billion. And every one of the state we talked with have a shortage. They want to use COVID drops because it's much easier, not getting into hospital, and a much small quantity. One run Regeneron can be 50 runs of Sorrento COVID drops because we're using about 2% of the quantity needed from regional quantity?
Sorrento, ticker symbol SRNE, one of my favorite companies, plenty of cash, big market cap. We're not talking about oncology. We're not talking about pain therapeutics, but we could do an entire panel just on oncology with Sorrento and certainly on the pain side as well. As you can see, nothing I like more than when the CEO is the scientist, right? Henry, I think your biggest issue is making sure you don't go into the lab and start kind of building, because we need.
I have my lab code behind me.
All right. So help me understand, how did you find Mark Emalfarb? Or Mark, how did you find Henry? How did you guys end up connecting with each other? And what are you proposing to do for Henry?
Go ahead, Mark.
Yeah, well, let me start out with, we found Henry because we have mined virtually everybody on the planet. We can think about where we can enable the production of larger quantities of lower cost vaccines, antibodies, and therapeutics. And Sorrento as an
Mark,
infrastructure.
Forgive me. Mark Emalfarb, CEO of Dyadic, ticker symbol DYAI, balance sheet, next event.
Okay, so balance sheet's 25.8 million in the last quarter, prior to the definitive agreement we're going to have with Henry, which will shore that up even further. $168 million market cap, 30% inside ownership, which is mainly me and my family, as well as some of the directors and insiders. No debt, no warrants, completely clean. And we're going to transform and revolutionize biomanufacturing.
Mark, are you dilution sensitive?
Yes, I'm very dilution sensitive.
You're the most dilution sensitive guy I know, and for investors, you should love that. Mark does not want to dilute his ownership stake, so he's not going to do any bad deals, right?
Well, we're only going to do tremendous deals with companies like Sorrento that have the power and the regulatory and the experience and the knowledge to pick the ball up and run it across the goal line. We've spent 25 plus years developing a way to mass produce proteins.
Okay, so I'm sorry I interrupted you. You found Henry.
Yeah, so we found Henry. We seeded Henry and his team with the receptor binding the main RBD protein of the SARS-CoV-2 virus. They ran a significant amount of animal studies, recognized in their lab the power and the potential of the vaccine itself in terms of its efficacy, its safety, its production. And so the combination between hyperproductivity and the effectiveness of not only the RBD from Wuhan, as Henry knows, we've made South Africa, UK, Brazil, and now Delta. So we have them all. And as you pointed out, and Henry pointed out, a traditional recombinant protein-based vaccine potentially has a safer, less side effects. And the RBD itself, like a flu vaccine, you take, it's quadrivalent. It has 4 different proteins. And I think, as you mentioned, Jason, the Wuhan strain, and as Henry mentioned, Pfizer, Moderna, J&J, et cetera, they keep giving you the exact same strain, the same antigen over and over and over again. I'm not saying it's bad, because it's better than nothing, but we're going to make a better vaccine in combination with Sorrento, we hope, where we're going to combine more than one of those multivalents. We could have 2, 3, or 4. And the ability and the technology that we avail over, that we're empowering Sorrento with, is to be able to not only do that, but to do it in large volumes affordably. We have the ability to treat a world population easily, simply, and affordably with a trivalent, tetravalent vaccine. That's being developed in Sorrento. We're doing it in Africa with the Rubik Consortium, in Korea with Meditox in Southeast Asia, in India with Syngene and Biocon. And with Sorrento, they have, in Israel, we're developing, that's what we developed. The base technology was developed with the Europeans and Zappi for 5 years before the pandemic. Somebody talks about, did we expect one? Guess what? We prepared for one, and we were ready. And with companies like Henry's and Sorrento's, they're going to pick up the ball and they're going to deliver the promise, and they're going to make vaccines, not just for America, but for a global population more affordably. And as you know,
Mark, if the Sorrento vaccine goes the distance, how do you benefit?
Well, we get milestones, up to $33 million in milestones, and we get royalties on each one of the vaccine shots per dose.
Okay,
so if you think about,
give me an example of that math. Not just kind of a generalized example, not guidance, but in a general way. Let's say Sorrento ends up, this becomes a popular vaccine and they do 500 million doses. You get 500 million times what? 10 cents?
Well, I'm not going to give the exact number, okay? But I think if there was an example given on our last conference call, if it's 0 to 10%, and if you just take something in the middle, 5%, and if you take 500 million doses, I think that adds up $250 million, drops to the bottom line with no cost of goods. If Henry does 1 billion doses, the number goes up exponentially.
Right. But my point is just that because we're talking about a lot of volumes, even though the net dollars per dose might be small, the reality is it always is in vaccines, and the secret is volume here.
Yeah, it's volume, but it's also, in the case of C1 and Dyadic, and it'll expand. I'm sure Henry has other thoughts and uses he's already thinking about of where he can apply this platform to make potentially antibodies or therapeutics or other drugs. We're not related or limited to COVID-19. It's a protein production platform that can be used to make dozens and hundreds of vaccines or antibodies or therapeutics. We're going to transform biologic manufacturing so that it's affordable and accessible. And as Henry pointed out, think about Regeneron and think about Lilly and the antibodies. The shortage is already in America. If you don't live in America, you're not getting an antibody for COVID treatment. It's not available, or it's not accessible, or it's not affordable. We can help solve that problem together with companies like Sorrento to really bring healthcare to a global population and health equity. The world is not going to stand for, let's say, part of the world getting vaccinated or treated any longer and the other part not. And we're going to help solve that problem.
So it sounds like over the next couple of years, as a vaccine comes to the market that includes the highly efficient production platform, the C1 platform, that could be transformative to Dyadic as it relates to COVID and as it relates to that platform, right? And that's one part of your business.
Right. It's only one part of a multi-pronged approach. We basically have an endless opportunity, but we're focused right now mainly on, to your point, Jason, therapeutic vaccines and antibodies and other treatments for COVID, but it's the opening door or the opening salvo to a much bigger picture.
Okay, so that's really interesting. And one of the things I didn't expect you to say was that, but it makes sense that you could manufacture therapeutics or monoclonal antibodies, the cocktail like COVID drops, more efficiently. Henry, is that something that you see too applicable to the C1 platform?
We're already doing it. We already received the antibody from Mark. We're testing it right now against all of the variants. If that works, we'll put it into a COVID shield, which is a cocktail of 2. We already have one from Mount Sinai, extremely potent, and it covers every one of the variant, Delta, Mu, Lambda, South Africa, Brazilian, you name it. And we have even tested dozens of theoretical mutants as a potential variant, evolving variant. It works on every one of them. Now, Mark just sent us with C1 produced antibody. We're going to test it. If that works, we have 2, maybe 3 antibody works on all of the variants. That would be amazing. Then it covers all of the bases, basically the whole 9 yard, right? From end to end, it covers all. And with a large quantity you can manufacture with C1 protein technology, then you really can supply the world. Since we're already using only a 1/50th quantity needed for Regeneron. If you can increase that quantity by 10-fold with C1 manufacturing, you're talking about 500-fold. Every time they run reactors, they run out of supply right now. Every one of the big pharma working with them, all of the raw material in shortage. You don't have supplies right now. Every state's knocking the door. They want it. They cannot get it. That's why they want to support the clinical trials with Sorrento COVID jobs, because then we can do 50 times the supply. Imagine if you do with C1, you can do 500 times the supply.
Mark, right? Sounds like Henry is drinking the Dyadic Kool-Aid. Sounds good.
No, we like it. We like it a lot.
Good. Let me get closing remarks from each of you. 1 minute or so. Amro, you've kind of heard the discussion from Henry talking about therapeutics, talking about COVID and talking about vaccines. We've had a little bit of a discussion about these are first generation vaccines. I love some of the comments, Mark, that you made when you're talking about the second or third shot of the same thing, which immunologically to me doesn't make great sense. And it's just sorting that out seems difficult. Maybe Aditxt is part of that solution. So close with us.
Jason, yeah, I mean, look, with what Henry and Mark talked about, ultimately right now, we have to understand whether it's a treatment or any other approach, it's got to begin with someone's immune profile. Aditxt could not be better positioned to provide that immune profile for COVID-19 and beyond, and many other indications. Ultimately, we as individuals and as a society will need to begin with understanding someone's immune profile. Beyond that, we didn't really spend much time on the therapeutic side to get into organ transplantation, autoimmunity, as well as allergies and immune tolerance and antigen-specific immune modulation. That's too much to put in all in one sentence. We could not be better positioned as we are looking to enter into clinical trials second half of next year, psoriasis, type 1 diabetes, skin allograft, and preclinical in multiple sclerosis. Could not find ourselves in better position.
Thank you. And Henry, 1 minute, give me a close. Boy, limiting Sorrento to COVID is almost criminal because there's so much else going on in the company, but it's also really amazing and very surprising to me how much you have going on just on the COVID side alone. Tell me a little bit about what's your message to people?
So my message is getting into commercialization. We have the cancer side with abivertinib, phase 3 done. We're going to be getting into data presentation very soon. And we will, if the data is good, which we see in the pretty tremendous phase 2 data, if it looks good, we're going to do pre-NDA meeting with FDA. That's on the cancer side. And non-small cell lung cancer, huge market. On the non-opium pain management, we've finished the 400 patient enrollment with SP102. We believe the peak sales about a $10 billion now because the drug shortage was dexamethasone oil for COVID patient use. And this could be a $10 billion product and no competition because no drugs are approved right now on this space. We have fast track. It's all done. We're going to submit hopefully next year. And COVID side, we about a commercialization on the COVID stick for quick detection. Then we get the large clinical trial done, proof concept, EUA approved COVID jobs could be a tremendous market opportunity. With if you get the different stage, we have MSC cells or oral pill with abivertinib to get you out of the hospital quick.
All right, thank you. And Mark, closing comments. Try to keep it tight because we're a minute behind, so you have 60 seconds.
All right. Well, again, I think our main goal, just like Henry said, we're going to bring healthcare, make it affordable, whether it be for rheumatoid arthritis, whether it be for cancer or diabetes. You think about Keytruda, they're selling Merck, $1.5 billion a month. We have to change the paradigm of the way these biologics are produced, or the world is not going to have access. And you know what? Working with Sorrento, working with Syngene, working with Meditox, working with Rubik, and we're working with other people on other products and other opportunities. The platform, when you put it in the hands of people like Sorrento and otherwise, or Syngene or Meditox or Rubik, they see the power, and they're going to start applying it in broader places and in diversity. And that's what we're going to do.
All right, guys, listen, this was a great panel. Thank you very much. I just want to remind everybody, if you're interested in Dyadic, if you're interested in Aditxt, if you're interested in Sorrento, go to the Dawson James website, pull down our research reports. You can see the initiation of coverage reports, and you can see the string of the most recent notes. They're open. They're available to everybody. Thank you so much.
Thank you.
Thank you.
Excellent conversation, gentlemen. Jason, you did a fantastic job moderating that. Thanks for all the good questions. And thanks for that little tip at the end on how we can find the notes. Appreciate that. You have a good day, sir.
Thank you.